thank you for the introduction, and it's really a pleasure to be here. Had an interesting day so far. So I'm going to talk about how chromatin structure influences genetic and epigenetic variation. And so here's an outline of my talk. So I was asked to give, like a little bit more sort of like a detailed introduction. And then I'm going to talk about chromatin and genetic mutations, and then some of our results concerning epigenetic changes. And then I have a small story. If there is time, we can talk about structural variation as well. But let's see. So introduction. So my background is in evolutionary genetics, and broadly I'm sort of interested in how properties of mutations influence evolution. And some of the questions we study in my lab currently is the sort of like rates and effects of effect distributions of epigenetic and genetic mutations, and their interaction sort of like between chromatin structure and genetic and epigenetic variation. And then recently we've started some new things looking at epistasis, but I'm not going to talk about that stuff today. But these sort of like different parameters of, like mutation rates and what are the sort of like effects of different mutations, they are the reason sort of like why I'm interested in these things is that they are sort of important parameters in many evolutionary models. And so this talk, going to talk about mutations, epimutations, and chromatin. So, and some of the questions are that we were interested in is how genetic mutation rate varies across the genome, and what are the factors that influence this variation. And also, how is mutation rate variation reflected on natural genetic variation that we observe. And furthermore, we are interested in do epimutations. So by that we mean mostly changes in DNA methylation, for example. Do they occur? Are they transmitted across generations? And what are these epimutation rates, and where in the genome do these things occur? But first, to sort of like understand this talk, you need to know something about chromatin, histone modifications, and DNA methylation. So here's a picture of DNA as it's shown in many textbooks. But in reality, like DNA is not sort of like floating free in the nucleus. It's wrapped around these protein complexes, which are called nucleosomes. And each of these nucleosomes is composed of many different proteins that are these different histone proteins. And DNA sort of like forms this structure that people usually think of, okay, this is like a string with beads on it. And string is the DNA and beads are these nucleosome complexes. So this is some, like something like the structure that DNA adopts in the nucleus. And then this chromatin in the nucleus is formed these different sorts of coils and loops. And some of the regions of the genome are more densely packed than others. And these densely packed regions are called heterochromatic regions. And those regions have a feature that transcription is usually silenced in these regions. And here is some more details about, okay, like how this packing works is that you have this beads on a string, and that sort of like loops and coils around each other to form these sort of more thicker fibers and so on. Some of the sort of like these things like how DNA is actually packed in the nucleus are a bit sort of like hypothetical at the moment, because figuring out what is the exact structure is actually a little bit difficult. So then these histone proteins, so they have this property that they actually have these sort of like tails that stick around from these nucleosomes. And if you sort of like zoom in on one of these tails, if we look at the individual amino acids that are in this sort of like tail of this histone, many of these amino acids can have some post-translational modifications. And the two modifications that I'm mainly going to talk about today are histone 3 lysine 9 methylation and histone 3 lysine 27 methylation. So H3K9 is a mark for heterochromatin. That's usually sort of like quite stable. It's called constitutive heterochromatin. And this H3K27 is a mark for facultative heterochromatin. So it's a different type of heterochromatin that is thought to be sometimes, like adopts a different conformation, is more active. And then you also have many other sort of these histone modifications that can exist, but we'll focus on these two for today. And then there is another region in the genome that is quite special. It's the centromere, where during mitosis and meiosis, the sort of like the spindle fibers attach and pull the sister chromatids apart. And those regions, they contain a special histone variant, which is called this SEMPA protein. Okay, so these are some of the modifications that occur. And then for those of you, if you are not familiar with DNA methylation, so this is basically a covalent modification of one of the bases in DNA, so cytosine in our case. So what happens is that the enzyme called DNA methyltransferase adds a methyl group to cytosine and modifies it. So you get this, what is called 5-methyl cytosine. And if you have this methylation, for example, in the promoter region of a gene, that usually silences transcription. And usually, DNA methylation occurs in symmetric sequence context. So, for example, in CG base pairs. So the idea is that the other strand, or both of the strands, are methylated. And when during DNA replication, when DNA replicates, it leaves this hemimethylated site, and these are also recognized by some enzymes in the cell, and they can, put this methylation back on. And this way, you can maintain sort of this methylation status across cell divisions. So, and these different sort of like chromatin modifications, they can affect how the chromatin is packed in the nucleus, and in turn affect like, okay, which transcription factors can bind to certain regions, and which genes can be expressed, and so on. And from an evolutionary point of view, the reason that I'm sort of like interested in these things is that because if this would be just some, like molecular biology of how, like transcription is regulated, maybe you could say that, okay, is it so important for evolution? But some of these chromatin modifications, we know now that they can be inherited. And if you have something like that can be inherited, then, like maybe that can be under selection or sort of like affect evolutionary outcomes. Okay, so just that I don't forget, I put the acknowledgement slide here first. So this work that I'm going to present has been mainly done with a former postdoc of mine, Mariana Villalba. And this research has also been funded by the Research Council of Finland. Okay, so back to the study questions. We're interested in sort of mutation rates and how it interacts with chromatin. And to sort of study these questions, we conducted what is called a mutation accumulation experiment with filamentous fungus Neurospora crassa. So Neurospora is sort of like the model system we use for a lot of studies in my lab. So it's this, I guess I would say, like old school genetic model organism. So if you remember this one gene, one enzyme hypothesis that Beadle and Tatum got the Nobel Prize for back in the day. So that work was done with Neurospora. So, but the reason we use it is that it has a small genome, so only 42 megabases, and it's haploid. So it has both asexual and sexual reproduction, and we can actually control the mode of reproduction by changing the environment. And more importantly, it has, like these different types of heterochromatin, which, for example, some yeasts don't have. All right. Then there is another sort of like relevant aspect of its biology that we need to talk about a little bit. So it has this system called RIP, which is repeat-induced point mutation. And this RIP system is, so it's present in some other ascomycete fungi as well. But what this system does is that during meiosis, it recognizes duplicated sequences in the genome, and it induces C to T transitions in those sequences. I mean, the biological function of this system is presumably to defend the genome against transposable elements or other sort of like foreign DNA. But it has a sort of like a consequence that over long evolutionary timescales, the genome of Neurospora crassa, due to the action of this RIP system, is such that it contains very few sort of like large perfect repeats. And the reason is that, like if you have some sort of like a array of repeated sequences, over time, RIP will induce mutations in these repeats. And so the repeats will sort of like diverge from each other. It has a quite low content of transposable elements. Only 14% of the genome contains sequences that have been annotated as derived from TEs. For reference, human genome has 44%. But I was recently told that this is also something that's quite close to Drosophila, so maybe not, like that unusual. But I guess sort of like what's important for this talk is that because this RIP system, like tends to sort of like make every repeat a little bit different, is that we can map short reads to centromeric and heterochromatic regions. And these can be sort of like done only with Illumina sequencing. For some other organisms, centromeric regions, for example, are extremely difficult to assemble because they contain these like large repeated sequences. But for Neurospora, we can do this. Okay. And so now I'm going to talk about this first part is about chromatin and genetic mutations. So this story has already been published. So if you are sort of interested about this, you can go on and read the full story. So we did this mutation accumulation experiment. And the idea in this experiment is that we take an ancestor, we split that into multiple lines, and then we propagate these lines by single spore descent. And that is sort of the key point of this experiment, is that we are only taking a single spore to the next transfer. And what this does is that it minimizes the effective population size within each of these lines. So selection against deleterious mutations is very inefficient. So this allows that, like even mutations that are deleterious, they can accumulate in these lines. Of course, we cannot remove selection completely. So if something is a mutation renders something like completely unviable, of course, that will be selected out from the experiment. And of course, the nice thing about Neurospora is that we can put these lines in the freezer. So we can start the experiment, put the ancestor in the freezer, do all of these transfers, and then at the end, we can take the ancestor out of the freezer and sequence all of these lines and compare them to each other and see what has happened. So, so the experiment looks something like this, is that we are going to plate some spores on a plate, isolate a single colony, put that into a tube and let it make more spores, then plate those spores again. And this is what we call one transfer. And in this experiment, we did 40 transfers in total. So the thing is that, okay, we want to calculate mutation rates eventually. But for this kind of filamentous organism, it's not immediately clear what is a generation. And because this experiment is, of course, like a little bit artificial in a way, the way we are growing it. So what we did is that we use a line where the nucleus has been marked with a green fluorescent protein, and we can count the number of nuclei. Okay, like if we have this much mycelium, how many nuclei we have and how many mitoses must have led to that number of nuclei. So we estimated how many mitoses happens in one transfer during our experiment. And so what we know is, okay, like during the whole experiment, each mutation accumulation line went through about 1,000 mitoses. So we started 2 independent MA pedigrees from 2 ancestors. So we have 40 lines in total. We do 40 transfers for about like 1,000 mitoses per line. Then we sequenced the genomes of these MA lines and combined this with some existing data where these different modifications in the genome are. And okay, we see about 33 mutations per line. In total, we observe some like 1,300 new mutations. Most of them are sort of like single base pair changes. We have these sort of like small indels as well, and then we have a few of these mutations which resulted in like change of like a 2 base pairs or something like this. But yeah, so mostly I'm going to focus on the single base pair mutations. So okay, first, so this is the 7 chromosomes of Neurospora. The sort of like blue lines show the chromosomes, and then underneath I've plotted some of these modifications that occur in the genome. So the gray boxes show the centromeric regions. These red boxes show regions where you have this H3K9 methylation, and these dark blue boxes show regions where you have H3K27 methylation. There's also this violet box, another histone modification, but that is actually like completely sort of like gives you the same information as the red one. So whenever this red one is present, the purple one is not. So sort of like statistically, the purple one doesn't give you any more information. Okay, and what we do is I'm going to overlay all of the mutations that happened in the experiment to this plot. So we see, okay, like where are the mutations that happen? And if you look at this, you already see, just looking at this by eye, is that you have more mutations in the centromeric regions. So that's quite easy to see. Okay. So we did a little bit more sophisticated analysis. So we use a model that accounts for differences in GC content. We also looked at, like if you account so for the flanking bases where the mutation happened. So these different sort of triplets, but the effect is more or less the same. And what we see is that mutation rate is higher in centromeric regions, but also in these regions where there is H3K9, and also slightly elevated in regions of H3K27. And this is sort of like taking the effect of GC content and some other things into account. All right. Okay. So if we sort of then like calculate mutation rate, so in euchromatin, mutation rate in Neurospora is like 2.5 times 10 to the minus 10 mutations per base pair per mitosis. But for H3K9 regions, this mutation rate is 10-fold higher. Okay, so next we sort of asked that, okay, we have these differences in mutation rate in the genome, but are those differences also reflected in the amount of naturally occurring genetic variation there is? So what we did is we used our model and sort of like put this genome into certain, like windows, and said that, okay, we use our model to predict the mutation rate for each of these windows in the genome. And then we use the data set of 57 genomes. So these are strains that have been sampled from nature and have sequenced genomes to look at genetic variation. And what we find is that, okay, we do see more genetic variation in these natural lines, in these heterochromatic regions, and also in the centromeric regions. And if we do this sort of like simple thing that we take theta, so the number of segregating sites, and we check that, okay, like how well it's correlated with our predicted mutation rate. Obviously, we see that there is a lot of variation in this kind of data, but we do see a relationship between our predicted mutation rate and the amount of genetic variation there exists in sort of like these natural lines. You might say that, okay, like in the end, this kind of correlation is maybe like not so impressive if you have an R square of 0.2. But of course, but then I would sort of say that, yes, but of course, like many other things affect natural genetic variation than mutation, like selection and demographic effects and so on. So I was actually like maybe a little bit surprised to see that this is as high as it is. Okay. So, but then of course, like somebody could ask like, okay, like what is the mechanism? Why do you have higher mutation rate in some regions than others? So we sort of have at the moment two ideas about this, like what could be the reason. It either could be that for whatever reason, some regions of the genome are exposed to more DNA damage, so some sort of like differential mutagenesis, or that the genome is repaired differently in these different regions. And we sort of like saw no sign that any sort of like common mutagenic pattern could explain these results. So because there are some mutations that should have some mutational mechanisms rather that have a characteristic mutational signature, like this deamination of methylated cytosines and these kind of things. But these mechanisms, like we cannot explain our results by those. And our hypothesis at the moment is that DNA is repaired less efficiently in these heterochromatic regions. And we think it's the reason is that this is our sort of like current model of how the DNA is packed in the nucleus. And for example, all of these centromeric regions of different chromosomes, they are sort of like clumped together and they are sort of like put in one place in the nuclear envelope. And then this euchromatin forms these loops in the nucleus, and some of these regions are then attached to the nuclear envelope as well. And and the idea is that some of these regions are sort of like packed more tightly, so DNA repair machinery cannot access them as efficiently as it can access euchromatin. Or the alternative is that DNA repairs could also be like specifically recruited to euchromatic regions. But this is something that we are sort of like currently testing if this is actually true or not. Okay, so summary so far is that we've seen that point mutation rate varies greatly across the genome, so order of magnitude higher in centromeric and heterochromatic regions. And we do see some relationship that if you have higher mutation rates, you also tend to have more sort of like natural genetic variation in those regions of the genome. But we still don't we don't have the mechanism for this yet. Okay, so then for the next part of my talk, I'm going to talk about these epigenetic changes. So we are going to look at DNA methylation. We are interested in, do spontaneous DNA methylation changes occur? Are they transmitted across mitosis? And what is the rate? And this work is currently unpublished. We are hoping to put the manuscript in bioRxiv very soon, I hope. Okay. So with DNA methylation, the idea is a little bit the same, but now we need to consider that these DNA methylation changes might have much higher rates than genetic mutations, especially if we consider what is known from other species, basically plants and Arabidopsis in particular, how this DNA methylation changes happen. And so we cannot ignore sort of like reversion or back mutation for these DNA methylation changes. So our idea, or the way we are looking at this, is that if we take samples from this MA pedigree, and then we say that, okay, for samples that are further apart in the pedigree, if we check where DNA methylation is in these samples and calculate some sort of like divergence in DNA methylation, if these DNA methylation changes are truly behave in a way that, okay, that they happen and they are transmitted to progeny, then divergence of the MA lines should increase over time. And samples like that are further apart in the pedigree should be more diverged in their DNA methylation patterns. We did whole genome bisulfite sequencing, so that is just a technique that can be used to sort of like reveal where are these methylated bases in the genome. So in our initial experiment, we took 10 MA lines from both of the pedigrees from 3 different time points. So ancestor, then after 5 transfers, after 20, and after 40 transfers. And looked at sort of like divergence in DNA methylation. We looked for DNA methylation changes both for single cytosines and then these differentially methylated regions. So the idea of or sort of like investigating these regions that change their DNA methylation together is sort of like related to the biology of how these methylation things are working. So normally if you have a single base that gets methylated, maybe that doesn't really have a sort of like a high biological impact. So some of these proteins that bind to methylated DNA, for example, they sort of recognize multiple bases. So we use this sort of like a hidden Markov type model to sort of like make the genome into like a small segments. And then for each of these segments, we ask that, okay, like does this segment have a high methylation or not? And has it changed in this experiment? So this is the way we sort of like classify these DMRs. Okay. And these are the initial results. So we looked at this and we sort of like immediately saw that, okay, this looks quite a bit different than, like what happens in plants. Because what we see is that, okay, there is maybe some initial divergence. If we look at these single sites, like maybe up to 400 mitoses, like divergence in DNA methylation increases, but then for sure there is some sort of like saturation in the data, and also the variation is increased, like really a lot. If we look at DMRs, the pattern is maybe a little bit more clear, but I wouldn't, like say that it's like super clean. I've just fitted, like a logistic growth curve there. I mean, statistically it fits better than, like a model with no divergence, but at this point this is a purely sort of like a phenomenological model, and I'm not inferring yet any sort of like rates here. Okay, so this was a maybe maybe something that we didn't like really initially expect. But then we said that, okay, what if we do the same and check where do these DMRs occur in the genome? And this is a plot again, neurospora chromosomes, and these black lines are now these DMRs. And we see that almost everything is in the centromeric and pericentromeric regions. We do have some DMRs outside the centromeres, but like clearly something is going on there. Okay, we saw this, and oh yeah, so one thing first. So yeah, we looked at this. So there are more DMRs in these centromeric regions than you would expect by chance, of course. So this is sort of immediately clear. So if we say that, okay, like, do the DMRs occur randomly in the genome? For sure not. They are depleted in euchromatic regions, overrepresented in centromeric regions, and also depleted in these H3K27 regions. So in plants, many of these methylation changes are often associated with transposable elements. So this is also like something that sort of like came to mind, that, okay, is this sort of pattern we see about these methylation changes that happen, does it have something to do with transposable elements? Because in these centromeric regions, there are a lot of sequences that are, like some old transposable elements or have been derived from such. So if we look at the number of DMRs that occur in the centromeres, many of them occur in sequences that have been annotated as TEs. If we look at euchromatin, most of them are in intergenic regions or even genic sequences. For these other heterochromatic regions, for H3K9, you also see that a lot of them are in transposable elements. But for H3K27, we see a little bit similar pattern than for euchromatin. But if we look at sort of like the overall picture, it becomes a little bit hard to explain that, like, is this only transposable elements? Because so if we look at these annotations a little bit more detail, in euchromatin, you observe DMRs in these transposable element sequences about, like the amount you would expect by random chance. And same for these H3K9 regions, and actually a little bit less for the H3K27. So it seems that, like, this is a little bit hard to explain by transposable elements driving this whole pattern. And we've done some additional analyses, and they sort of like, we don't sort of like see something that, like we could neatly explain this by TEs. Okay, so, but then we thought that, okay, if all of these DMRs mainly occur in the centromeres, what if we check the divergence again, but now separating sort of the different regions of the genome? And what we noticed that it's only the DMRs that occur in the centromeric regions that we have any evidence that they are transmitted. So DMRs that occur elsewhere, so there seems to be like no divergence over time. So either they are not transmitted across mitosis, or they are, like so unstable that they disappear so fast that we cannot like really see that. Okay. So at this point, we were sort of like scratching our heads and saying that, okay, are we looking at something that is a biological phenomenon? And to convince ourselves, so we sampled another set of these MA lines from the 2 pedigrees. And this time we use nanopore sequencing to detect DNA methylation. So without going into details, it's another sort of like technique to detect DNA methylation by sequencing. And the sort of like idea here is that, okay, just to be sure that like there isn't some like technical issue with the bisulfite sequencing that would cause us to detect more methylation in certain regions of the genome. And we also wanted to have more denser sampling. So now we had 6 time points, and those were separated by a lot fewer transfers. So we had samples after 1, 5, 7, 8, 10, 15 transfers. And hopefully, like if it's really the case that these ML lines are diverging from each other, if we have a more denser sampling, maybe we now sort of like catch the linear divergence better. And we replicate the result that divergence in these methylation patterns happens only in centromeric regions. And for the nanopore data where we have denser sampling, it's indeed true that now the divergence is much more linear. Okay, and now that we had like this kind of observation, so here we could actually fit like a sort of like a more mechanistic model of this sort of like a epimutation accumulation. So we could estimate sort of the rates of gains and losses for these DNA methylation changes. Yeah, so, and I should say that the model of neutral accumulation fits much better than sort of like the null model of no accumulation at all. Okay, and here is just some examples. I didn't put all of them to the slide, but what we see is that gains of methylation occur at much slower rates than losses. So losses of methylation is about 50 times more common. And these rates seem to be quite similar for DMRs and single sites. And these rates, of course, like if you have a gain of methylation, something like 9.5 times 10 to the minus 6. So that's many orders of magnitude faster than the rate of genetic mutations. Because one could also ask that, is it possible that, because in these lines there are these genetic changes, so could these genetic changes somehow explain these DNA methylation changes as well? But so far, we cannot really explain them by genetic mutations. So first of all, because the rate of these methylation changes is so much faster, and we have so much more of them than genetic mutations. So you would have to assume, I think, some like really strange patterns that if you have a genetic mutation, it changes, like a bunch of DNA methylation in other chromosomes in a very sort of weird way. So we don't really see that, like we could feasibly explain this phenomenon by, like genetic mutations. Okay. So what we see is that, okay, we have these spontaneous changes in DNA methylation that are transmitted across mitosis. And these rates of DNA methylation are sort of changes are orders of magnitude faster than for genetic mutations. But these seem to occur only in the centromeric and pericentromeric regions. And we don't have any evidence as of yet, at least, that it's transposable elements that are driving this pattern. It seems to be some other property of the centromeric regions. Then, because we did observe these sort of like very transient methylation changes outside the centromeric regions. So we had, like DMRs were sort of like enriched in genes that are in these H3K9 regions, and for example, and depleted in genes in euchromatic regions. But it seems that these kind of changes, there seems to be like quite limited scope for regulating transcription if, like they are lost quite fast. And this was also like quite puzzling for us at the beginning, but now we have a hypothesis about this, and there is a specific pathway in neurospora that's also like related to silencing of like some foreign DNA like transposons and viruses, for example. And so the hypothesis is that so it's a pathway called ATRX, so that this ATRX system is sort of like scanning the genome, and these transient methylation changes we observe outside the centromeres are sort of kind of like a byproduct of the action of this pathway, as it's sort of like checking that is there something strange going on. But this is at the moment, of course, something that we need to test. All right, I think I have a few minutes. I'm very quickly going to go sort of like over some very recent results. So we've also done we've been looking at structural variants. So this is together with my another postdoc, Snøfrid Kleppe, and we have a sort of like set of genomes that we've sequenced both with Illumina and with nanopore, so to get long reads, and so to call like both SNPs and these sort of like larger indels. And what we find is that, okay, structural variation seems to be a little bit different across the genome. So if you look at chromatin, you see, like a kind of enrichment of these very small structural variants. And but overall, you find less structural variation in chromatin than you expect. And centromeric regions and these H3K27 regions are again enriched. But what was a little bit surprising that these H3K9 regions that have a higher mutation rate for point mutations, at least, they also don't have so many structural variants. And so this is some preliminary analysis. If we split the genome into sort of like small bins and calculate theta, okay, like how much variation there is. Looks something like this for SNPs and structural variants. So we obviously have a lot of these bins where there's no variation. So we use this sort of like a hurdle gamma model to take into account that there's a lot of zeros in the data. And what we see for the SNPs, that for SNPs, there is a more variation than in chromatin in H3K9 regions. But for structural variants, this is reversed. So there is more structural variation in chromatin than in H3K9. And this we definitely need to investigate further, but we are sort of like thinking about is that some of these H3K9 regions, the sequence of those regions itself is not important, but it's important that you have this heterochromatin for some sort of like structural reasons. So without it, the genome is sort of like unable to function probably, and this is a reflection of purifying selection, sort of like acting to keep the heterochromatic regions there, even though their exact sequence is maybe not so under strong selection. But this is definitely something that we need to think about further. Yes, I think that was my talk, and I'm happy to take questions.